Intriguing tone. The human cornea is a structural anomaly, a living, biological tissue that allows light to pass through virtually undisturbed. Millions of people require corneal transplants, yet standard 3D bioprinting methods often produce cloudy, unusable results. Standard bioprinting extrudes collagen in a disorganized web, causing light rays to scatter instead of reaching the retina. Researchers found a way to resolve this by utilizing the fluid dynamics of the printing nozzle to align these microscopic strands. Transforming a cloudy gel into a clear optical window requires precise control over the mechanical forces acting on the ink. The process begins with a specialized bioink composed of collagen fibrils and living keratocyte cells. As the ink enters the nozzle, it experiences friction against the walls, a force known as wall shear stress. This stress acts like a microscopic comb, pulling the molecules into a single direction. The immense extrusion pressure forces these chaotic fibrils into a highly organized, parallel alignment as they exit the tip. The nozzle geometry dictates the architecture of the tissue before the ink even reaches the surface. However, the magnitude of this pressure is critical for both clarity and cell survival. A wide nozzle creates insufficient shear stress, leaving the collagen in a disorganized, light-blocking state. Conversely, a narrow 30-gauge nozzle achieves high alignment but subjects the living cells to lethal levels of stress. This trauma forces the cells to produce ACTA2, a genetic marker for the opaque scar tissue that destroys transparency. The 25-gauge nozzle provides the middle ground, enough force for alignment without triggering the scarring response. Hitting this mechanical threshold is the difference between a viable transplant and a blind scar. The initial printed alignment serves as a guide for the cells to begin remodeling the tissue. Over 28 days, the cells follow these tracks, growing in a structured, directional orientation. These cells eventually secrete new collagen perpendicularly, creating the crisscross lattice found in a healthy human cornea. Without the initial shear stress to guide them, the cells simply reconstruct a random, opaque matrix. The printer's mechanical force effectively directs cellular behavior toward mimicking natural human tissue development. The final validation is whether light can pass through this engineered lattice without scattering. Transmittance data shows the 25-gauge print nearly matching the clarity of native human tissue, achieving nearly 100% transmittance at visible wavelengths. In vivo tests confirm the result. A 25-gauge cornea transplanted into a rabbit eye remains clear enough to see the pupil below. Meanwhile, the non-printed control results in a milky, obscured implant that blocks vision. By mastering microscopic fluid dynamics, we have a reliable method to print clear, functional replacements for the human cornea.